Hello, good afternoon, everyone. Let's get started. We are going to be doing lecture 8 this week. The first half of lecture 8 is mostly um, theory stuff, and then the second half is more application problem solving type of discussion. Okay. And the topic is heredity. So let's start by talking about meiosis. The purpose of meiosis is to create reproductive cells so that we can have sexual reproduction. Right? Previously, we learned about mitosis, which is just a single cell dividing itself, creating two identical copies. And for some organisms, they use mitosis as a way of asexual reproduction. However, in humans, we do sexual reproduction, and that involves creating these reproductive cells where each reproductive cell contains one set of the chromosome, or one set in the sperm, one set in the egg. And when they come together, you will recreate the full set for the next generation. So meiosis is the basis of sexual reproduction because it's the process by which we create the sperm and the egg. So there are some new terms that I need to introduce to you this week, starting with haploid and diploid. Okay, so we use the letter N to represent one full set of chromosomes. So in humans, the N number is 23. Our somatic cells are bodily cells. These are like liver cells, muscle cells, brain cells, heart cells, all our regular bodily cells. They are diploid, meaning they contain two sets of chromosomes. So they are 2N, okay, two sets of chromosomes. 2N is basically 2 times 23, which gives us the 46 chromosomes that we are familiar with. In the process of meiosis, the diploid cell is going to divide, and in doing so, it causes the genetic material to be reduced by half. So now the egg has one set and the sperm has one set, and we call them haploid. When fertilization takes place, when the sperm fertilizes the egg, then we recreate the diploid cell, and then that propagates to the next generation. Okay, so that's haploid versus diploid. Here is another example. Okay, over here, one set, which is haploid, contains three chromosomes, the pink one, purple one, and the yellow one. So this is N equals to 3. If we are looking at a cell that is diploid, then in this case, it would be 2N, which equals to 6. There are 6 chromosomes in total, and there are two full sets. Two pink one, two purple one, and two yellow one. Okay, so once again, we are diploid, but not everything is like that. Okay, there are organisms that are triploid, meaning there are 3 sets of the chromosome. So here, 3N equals to 9. Right? So basically, this coefficient, right? This number in front of the N, that tells you how many sets you have. And then this here is how many chromosomes you have in total. Right? So over here, 4N equals to 12. Strawberries has 8 set of chromosomes, so 8N. I don't remember off the top of my head how many chromosomes they have in one set, but they have 8 sets of it. And yam or sweet potato, they have 6N. So anytime you have more than two, then it's called polyploidy. Polyploidy. Okay, you can call them tetraploid or octoploid, but overall they are all polyploid. Okay, polyploidy. Now, one of the questions that really bothered me when I was learning biology at a younger age is like, how do they make seedless watermelon? Okay? So to grow a plant, you need a seed, but if the watermelon does not have any seed, how do you make more seedless watermelon, right? Well, it turns out it has to do with these polyploidy concepts. Regular watermelons are diploid. That means they have two sets of chromosomes. Again, this only tells you how many sets it has. It doesn't tell you how many chromosomes in each set, right? Now, there's a special breeding technique that we're not going to talk about, but it allows the farmers to create tetraploid watermelons. Okay, so these watermelons that are specially bred has four sets of chromosomes. And then what happens is they cross means they breed. They cross the regular watermelon with the tetraploid watermelon. So if you have been paying attention, can you help me out a little bit? When this thing produces the gametes, the reproductive cells, okay, how many sets of chromosome is it going to have? Gametes are, like sperm and egg, but for watermelon, it's like pollen, right? Pollen and egg. So how many? Yes, just N. Thank you, Cheyenne. And what about this one? What about the 4N watermelon? When it produces the gametes, then how many sets of chromosomes is it going to contain? Two sets, that's right. So for those of you who might be wondering like how to get these numbers, you basically divide it by two, right? Because the process of meiosis is going to reduce the number of chromosomes by half. Okay? So if you breed these things together, N plus 2N, it's going to create a 3N watermelon. And it turns out the offspring that has three sets of chromosomes, they are actually sterile and they do not produce any seeds. And that's how they make seedless watermelon. So the important point here, in case you miss it, it turns out cells with odd number of odd number set, odd numbers set of chromosomes cannot undergo meiosis. Okay? And if you cannot undergo meiosis, then you cannot create gametes, no babies, right? No seeds. So it should make sense to you why odd number sets cannot undergo meiosis because meiosis divide the number of chromosomes into halves. So if it's an odd number, you can't divide by half. You're not going to get a whole number, right? 3 divided by 2 is 1.5. You can't have 1.5 sets of chromosome, right? You're going to be losing some of the genetic material, right? So that's the takeaway point, okay? In this slide. Are we okay? All right. So a couple more words, new words for you. The process of sexual reproduction begins with meiosis in germ cells. Okay, so in case you forgot, germ cells are diploid, they're 2N, and they are able to undergo meiosis and create gametes. Okay, so gametes are sperm or egg, and this takes place in the gonads. Okay, gonads are reproductive organs. Okay, so for men, the gonads are testes, and for female, the gonads are ovaries. And that's where meiosis is going to take place. And it's going to be generating the gametes. Once the sperm fertilizes the egg, then it forms a zygote. Okay? A zygote is diploid. Right? The sperm has already inserted the genetic material into the egg, and now you have two full sets. And that is the first single cell that essentially we all came from. Right? So that zygote will rapidly divide in the following days, and it becomes the embryo. Right? It will implant itself into the uterine lining, and it will grow and become the fetus and baby eventually. So here's an overview of meiosis. You start with a diploid cell, which is 2N. For simplicity, we are only showing a pair of chromosome here. So if you count the chromosomes here, that's one chromosome, two chromosomes. Okay, two chromosomes. And then you will copy the DNA. So now it's duplicated. We have the sister chromatids, and then it's gonna separate. And now if you count the chromosomes here, because the chromosome is stuck together, we just count it as one, right? So you have one chromosome here, one chromosome here. So after the first division, the amount of genetic information is already reduced by half. And then you break this sister chromatids apart when you do a second round of division. And if you count the chromosomes here, it's still one, right? So it is haploid once again. So you start with a diploid cell. After first division, it's haploid. And then after the second division, it is haploid again. So this is a little bit confusing to look at, I suppose. So what we're going to do next is we're going to compare mitosis and meiosis. Okay, this is in your study guide. If you have it, you can follow along. If not, just make a chart on piece of paper, and then we'll fill it in together. So let's start with mitosis, right, which is what you've learned last week, right? You had a quiz on it. The quiz was very well done. The class average is like, I think it was like 89 or 90 or something. That was something ridiculously high. So well done. And which means you should be familiar with it. Okay, so mitosis, the number of cell division. Okay, so you start with one cell, and the cell is basically going to divide, and then afterwards you end up having two cells. Okay, so number of divisions, one division. Okay, how about meiosis? Well, as you just saw on the slide, meiosis will divide twice. Okay, so first the cells split, and then you have two cells, and then they will split one more time, and then you end up having two cells. So this would be two divisions. Okay. How many daughter cells do you create in mitosis? Two daughter cells, that's right. Okay, two daughter cells. And for meiosis, if you count from the picture, it's four, right? But actually, it's a little bit different, okay? So for male and female. So for female, the meiosis is gonna give you one egg plus three polar bodies. I'll explain what that is, what these polar bodies are later, okay? Just write them down first. And For men, for men, for male, the meiosis is going to create four sperms. Four sperms. Okay. We'll come back to that later. In both cases, for mitosis and meiosis, we start with a diploid cell, which is 2N. What can you say about the daughter cells? Are the daughter cells diploid or haploid? Diploid is correct. Thank you. Okay, we're talking about humans here, right? Humans. So diploid is 2N. And for meiosis, after the first division is already haploid, and when it divides again, it is haploid again. It's not this is confusing for some people. Like they don't understand like why okay, 2N divided by 2 is N, and then when you divide it again, how come it's not like N over 2 or something like that? It's not like math necessarily, okay? It's just the way we count the chromosomes, right? So if you go back to this picture here, when they're stuck together, we count it as one, and then after you break it, now we count each individual piece as one, right? It's like that Kit Kat analogy I told you about, right? When they're together, you count as one. Once you break it, now I have, two separate ones. So that's that. Okay, number of chromosomes in daughter cells, 46 and 23. That's correct. Okay, prophase in mitosis. Let's draw it out. Okay. Hopefully you have two colors. Okay. So in prophase of mitosis, you're gonna have, chromosome. For simplicity, I'm just gonna draw two chromosomes, one big and then one small, okay? So that's chromosome one, and this, let's just call it chromosome two. And of course, you're gonna have another one. Um, I just want to ask to see if you guys, oops, sorry, to see if you remember, what do we call these chromosomes? The big red and the big blue. They have a relationship. We have a name for that relationship. Starts with an H. Anybody? No? Yes, homologous. Although I think you spell homogeneous, which is like chemistry, but this one is homologous, okay? Same similar idea. Homologous chromosomes. Okay. Yeah, that's all right. Homologous right here. Oops. Okay. So in mitosis, homologous. I'm gonna zoom in a little bit in case some of you are watching this on your phone, which can barely see what I'm writing. Homologous chromosomes are randomly, randomly located throughout the cell. Okay. You just see them like here and there, all over the place. So far, nothing is new. Everything is things we've talked about. And then we're gonna have a new thing over here. Okay? This is prophase one. I probably should explain a little bit more. Um, so when you have a cell and then it's gonna do meiosis, it's gonna divide once and then it's gonna divide again, right? That you already know. So this first part, it is called meiosis one, and in meiosis one, it has PMET one. So prophase one, metaphase one, anaphase one, telophase one. And then over here, this is meiosis two, and you have PMET two. Prophase two, metaphase two, anaphase, and telophase two. Okay, so here we are comparing prophase one with regular mitosis prophase. So on the left side, this is what happens in mitosis. We learned that last week. Now we're going to draw what happens in prophase one of meiosis. And please use color, otherwise the note will not make sense. So in prophase one of meiosis, homologous chromosomes pair up to form, and here is another new word for you, to form tetrad. So tetrad means four, and here is a tetrad. And here is another tetrad. Okay, so they will find each other. Chromosome one will find the other chromosome one, they'll pair up. Two will find two, 20 will find 20, so on and so forth. So you're going to have 23 of these tetrads. If it's female, okay, the X will find the X. For male, you will only have 22 of these tetrads. The X and the Y are not homologous. Y is much shorter, so they won't find each other. Okay. But the point is they will form these tetrad, and the purpose of forming these tetrad is they're going to do something called crossing over. Okay. So here is a crossing over. Here is a crossing over. It's literally crossing each other over, okay? And when they cross over, it allows exchange of genetic information between the mother chromosome, the maternal chromosome, and the paternal chromosome. Okay, and this is happening inside your cell, right? When you produce gametes. Okay, so you inherit the red copy from your mom and the blue copy from your dad. And now when you are producing reproductive cells, you are swapping genes between those two copies, shuffling the genes essentially before passing it on to the next generation. Okay, so this is important. Okay, this is important for generating genetic variation in gametes. Okay, so let me draw it out for you a little bit better to help you understand. So this is the mother chromosome in your cell. then the paternal one, and then after crossing over, you take a look first, please. You can basically, okay, a little bit of the blue will swap with the red, and now it's like this. That's a wrong blue, I think. There you go. Okay. And then similarly on the other one, you're gonna swap. Right? So these two parts gonna exchange genetic information. Right? So now it's a mix of red and blue. Now this is an oversimplification, okay? In reality, you're gonna have like mix along the entire uh, chromosome. So in the end, it's like re completely reshuffled between the red chromosome and the blue chromosome. Okay, and you get something like that. Okay? But it's like difficult to draw, right? So that's why I'm just showing you like a portion of it. Okay. Do you understand this situation? Anybody want to ask anything? It's like if I have a deck of red cards and then I have a deck of purple cards, okay? And then I'm swapping the cards, okay? So like if I take like a ace, okay, from the red deck, I have to swap it with an ace from the purple deck, okay? I cannot take a two and swap it with a six, okay? You have to exchange the same genetic information. So, if it's eye color over here, you are switching for eye color on the other chromosome, right? And then you do that throughout the whole deck. In the end, instead of having the original red deck and the purple deck, now each deck is a mix of red and purple. Does that help a little bit, maybe? Okay, so this only happens in meiosis, okay? Because we're creating genetic variation in the gametes. So our kids are not going to look exactly like us. Okay, we do not want this in mitosis because mitosis, we want a faithful copy of the original cell. When our skin cell divides, we want all the skin cell to be identical, right? Otherwise, it's going to create a mole, or worse, we're gonna have, maybe uncontrolled cell division, right? All right, moving on to metaphase. First, let's review what happens in mitosis. In mitosis, metaphase, the chromosomes line up at the center. Okay, you can draw the centrioles. I'm just gonna put a label there for you. Oh my goodness, what did I press? Okay, that's the centriole. And then the spindle fiber is coming out, right? So chromosomes line up at the center. Yeah, at the metaphase plate, if we want to be, proper about it. But yeah, I'm just gonna wait, line up at the middle of the cell. Yeah, at the middle of the cell. Okay. I'll give you a moment to catch up. So over here in metaphase um, one of meiosis, Okay, now remember, crossing over has already occurred. So, the red is going to have a bit of blue and the blue is going to have a bit of red. And again, I am emphasizing the fact that this is oversimplification. In reality, well, first of all, in reality, they are not red and blue. Okay, they're all colorless. Uh, but the exchange would have occurred throughout the entire chromosomes. Okay, so it's really a blend of blue and red all the way across. Okay, but the key thing here is instead of chromosome lined up at the middle of the cell, right? The tetrads line up at the middle of the cell. Okay. Now I'm gonna add a little thing here. The orientation. Okay, the positioning. That's what it means, right? The positioning, the orientation of each tetrad is independent of each other. This further increases the genetic variation. Okay, let me explain first before you copy. So for the first tetrad here, we have the red chromosome on top and the blue at the bottom. And that's completely random. It could have been blue on top and then red at the bottom, right? And then the next pair has blue on top and red at the bottom. So whether it is red on top or not for the first pair has nothing to do with whether it's red on top or not for the next pair, right? So each of the tetrad is going to randomly has its own positioning. So it's like flipping a coin 23 times and then, you're recording the result. And in the end, you will get a set of a randomized positioning of all the tetrads, and that creates additional um, genetic variation, right? So like you have two choices for the first one, two choices for the next one, and then two choices. So this is basically like 2 to the exponent 23, right? Different ways of arranging the chromosomes. Does that make sense? Okay, let me know when you're ready, and then I'll move on to the next part. Just give me a sign. Only 9 people here today. Not sure where the rest of the class is. All right. Almost there. Let's take a look at anaphase. Okay, this is the last thing that's gonna have a bit of a difference between the two. So for anaphase, we are going to again draw the centrioles. And can somebody tell me what these things are? The ones I'm drawing right now. What do we call them? They were stuck together before and now they're separated. Do we have a name for them? That's correct. Thank you. That is the sister. These are the sister chromatids. Okay. So in anaphase, sister chromatids are separated. And pulled to opposite ends of the cell. Over here, instead of sister chromatids separating, what should we say? What's the relationship between these things? When they used to be together as a tetrad and now they get separated. So what should we say? Okay, so they are homologous. Homologous chromosomes are separated and pulled to opposite ends of the cell. Okay, so these are homologous chromosomes. All right. So this is going to split into two, and one cell is going to get this, and one cell is going to get that, and that would be telophase 1. Now let me show you the slides just to let you see what it looks like. So this is prophase number 1. We have the tetrads forming, and you can see the crossing over occurring. And then the tetrad lines up in the middle of the cell. We pull apart the homologous chromosome, and then you're going to get two separate cells. Okay? So these two chromosomes on the top, like purple, purple, blue, blue, purple, purple, blue, blue, right? And then at the bottom, we have the opposite. Okay, the color is not as important as the size, right? At the beginning, we have one big and one small, right? So you have to make sure that each cell is going to have one big and one small because it's chromosome 1 and chromosome 2, right? You cannot have both of them as big chromosomes. Then you end up having extra chromosome 1 and missing chromosome 2, right? And then meiosis 2 is exactly same as mitosis. So we have prophase again. The individual chromosomes line up in the middle. You separate the sister chromatids. And then now you're going to split the cell. Split the cell. So that's going to be one cell, second cell, third cell, fourth cell. And then you get four cells that are genetically different from each other. Okay? And one more time, it's not actually blue and purple. The blue would be mixed with purple along the whole length, and the purple would be mixed with blue along the whole length because of the crossing over that occurred all the way back in prophase number 1. Okay, so let's complete our note here. Genetic relationship of daughter cells to the parent cells. For mitosis, they are identical. For meiosis, they are not identical. Type of cells that are involved in mitosis would be somatic cells. And for meiosis, it's going to be germ cells in the gonads. Why do we do mitosis? For growth and development. Replace, loss, or damaged cells. And for some organisms, they use mitosis for asexual reproduction. But since this chart is about humans, we're not going to put it there. For meiosis, the purpose is to form gametes for sexual reproduction. Okay. So I'm not going to ask you to describe meiosis, but you should, study this chart to make sure you understand key differences between mitosis and meiosis. All right, let's do a few more slides before we have a break. Gametogenesis. Genesis is creation, gameto, creating gamete, basically, right? Gametogenesis is the creation of gametes. And like I said, gametes are cells involved in sexual reproduction. It takes place in the testes for male and ovaries in the female. For male, the type of gametogenesis is called spermatogenesis, creation of sperm. And the process is normal meiosis. For female, it is oogenesis. That's not a typo, it's actually double O. And that is the production of egg in the ovaries. And the process is a little bit more complicated compared to spermatogenesis. And I will show you the difference. On the left side, we have spermatogenesis, which begins with the germ cell. This is the germ cell called spermatocyte. So you start with 46, which is 2n. And after the first division, how many chromosomes in the cell? What number should I put here? Just to check if you guys are with me. 23 is correct. Okay, you already cut it in half. So this is haploid, and then it's going to be haploid again. So it creates 4 sperm cells, right? Normal meiosis. For female, oogenesis begin with a oocyte. This is the female version of the germ cell. And 23, 23. What we have here is uneven cytoplasmic division. One cell gets a majority of the cytoplasm, and then the other doesn't really get any. And this is important for forming the egg because the egg has to supply all the nutrients and all the organelles associated with creating the zygote. The sperm only contributes the DNA, right? And so the egg needs to be substantially bigger. And so we cannot split the cytoplasm 50-50. Right? One has to get all of it. The one that does not get too much cytoplasm is called the polar bodies, and the polar bodies is not going to become the egg, basically, right? So this is reabsorbed by the body. Sometimes the polar body can divide again and you generate additional polar bodies. And over here, when this cell divides again, we have another round of uneven cytoplasmic division. So that's why you can have three polar bodies. Okay, none of them are going to be eggs. Only one egg in the end. So that's the first difference between spermatogenesis and oogenesis. Okay, so the first couple points we already talked about. Take a look at the last point here. For males, the sperm production process is continuous. Okay, after puberty, the testes continues to produce sperm up to very old age in many cases, and the production is very high, about 200 to 300 million sperms per day. And if you do the math, that's roughly 2,000 to 3,000 per second. But only about half of these becomes viable. We'll learn more about sperm production in second semester, okay, when we do the reproductive system. For females, it is a little bit different. The process is discontinuous. So actually, when the baby is in the mother, if it's a female baby, um, the process of meiosis already began even before birth. Okay, but the oocyte is going to be suspended. It's going to be arrested in prophase number one. Okay, so it's going to do the crossing over, and then it's going to stay that way until puberty. After puberty, um, each cycle, like roughly once a month, but obviously can be longer or shorter depending on the person. Then one egg will be selected and it will continue to go from prophase to metaphase 2. And then if fertilization takes place, then meiosis 2 will complete. Otherwise, the egg is not used and it leaves the body through the menses, right? So a female is born with all the eggs that they're going to have in their lifetime, okay? Which is more than what one would need, okay? Not all of them is going to end up, progressing through the entire process. Um, it's quite a large collection, right? But because you're born with all the eggs you're gonna have, the quality of those eggs tend to deteriorate over time. And so women who decide to have child later on in life are at a higher risk of having a child with a chromosomal disorder. And also, it might be more difficult to have child because of the quality of the eggs later on in life. So one of the problems that can occur is what's called non-disjunction. Non-disjunction. So non-disjunction is the failure for chromosomes to separate in either anaphase 1 or anaphase 2. Right? These are the consequences. It's much easier when I show if I show you it to you on a picture. Yeah, so let's take a look at um, the situation here, okay? Non-disjunction in meiosis 1. For simplicity, I am only showing you a tetrad. Okay, so these are all the same chromosome, chromosome 1111, for example. And over here, that is the first meiosis. Okay, so we are focusing specifically on anaphase 1, where the homologous chromosomes are supposed to separate. And it does. And you have the homologous chromosome separating here, no problem. And then we're gonna have anaphase 2, okay, which is when the sister chromatids separate. So if they do separate, then no problem. We end up having cells that would be haploid. And if a sperm, which is also haploid, fertilizes these eggs, then you will have 2N and 2N. So this is normal. However, if non-disjunction occurs in anaphase 2 and the sister chromatids fail to separate, then one cell will get both of them, and then the other will have none. So this would be n plus 1, whereas this is n minus 1, meaning this cell would have the full set of chromosome and an extra one. This cell would have the full set minus the one that's missing. So if the sperm fertilizes this egg, remember sperm contributes N, what would be the N number for this cell? Like think of math class, right? Okay, like this is N plus 1, this is N. So what's the final expression here? Yes, thank you. 2N plus 1. Okay, 2N plus 1. Meaning this zygote, which will become the baby, has two full sets, one set from sperm, one set from egg, and an extra one. Okay, so this creates trisomy. Okay, trisomy. Depending on which chromosome is extra, then it could be like trisomy 18, trisomy 21. Right? Trisomy 21 is usually the one that people are familiar with, right? Which is Down syndrome. Okay. Over here, this would be 2n minus 1. So these are usually non-viable. Okay. The baby will not develop to full term. Okay. So if non-disjunction happens in anaphase 2, 50% of the egg will be okay, 50% of them will be not okay. And since anaphase 1 happens each month during the cycle, the later on you wait for trying to get pregnant, for example, then those eggs would have been sitting there for a much longer time, right? And the chance of having these non-disjunction problems increases exponentially. If non-disjunction happens in anaphase 1, then it's even worse because now the homologous chromosome fails to separate. So this cell has extra, this cell has missing. The cell that's missing will now produce two additional cells that are missing the chromosomes. So 50% will be 2n minus 1, non-viable. And then 50% here will be 2n plus 1, which would lead to, again, different kinds of chromosomal disorder. All right. And depending on which chromosome is extra, some forms are more severe than others. And the outcome and the prognosis would be different as well. Does anybody have any question about non-disjunction? Okay, so obviously this is important information for a mother to know, right? To be able to know if the baby they're carrying has any kind of genetic abnormalities. And one of the ways to check this is to prepare a karyotype. A karyotype is basically an orderly arrangement of the chromosomes taken from the cell of the person you want to look at. Um, and and once you extracted the DNA, you can look at the DNA under the microscope, and the computer will take a picture of the chromosomes, and then the software will arrange the chromosomes in order. So chromosome 1 is the longest one, and then chromosome 2 is the second longest, so on and so forth, right? This particular slide is XY, so it is a male that we are looking at, and you can see there are no extra or missing chromosomes, okay? Now, how do you get the baby's cell? Well, there are two methods that are being used. One of them is called amniocentesis. Amniocentesis involves inserting a needle through the belly button or around the belly button and into the womb. This process is guided by ultrasound. So, you can't just randomly stick a needle in, the baby is in there, right? Like you have to at least have some way of seeing where you're poking. So you stick the needle in and you take a little bit of the amniotic fluid, which is the fluid that surrounds the baby, protects the baby while it is in the womb. That fluid contains cells that got washed off from the baby. Okay, so once you take a collection of the amniotic fluid, you retrieve the cells from it, and then you extract the DNA and you prepare a karyotype. Okay, in this case, you can see this particular karyotype shows there is an extra chromosome 21, meaning the baby has trisomy 21 or Down syndrome. Amniocentesis can be performed between 15th to 20th week of pregnancy. So it's relatively early in terms of the whole pregnancy. But if the mother wants to perform it even earlier because she is at a higher risk, for example, of carrying chromosomal disorder for her child, then they can do another technique called the CVS, which is chorionic villus sampling. The chorionic villus is basically a little bit of a projection extension of the placenta. The placenta is what connects the mother and the baby, right? So the placenta tissue actually has the same kind of cell that the baby has. So in here, it's a transvaginal sampling and goes through the cervix here, and you take a little bit of sample from the chorionic villus, and from that, you will extract the DNA and prepare our karyotype. This can be done as early as 9 weeks in pregnancy. Okay. Of course, these procedures are not without risks because you are, entering the womb one way or another with instruments, and that could sometimes harm the baby in an unintended way. But nevertheless, they give valuable information about the baby's genetic profile, and it allows parents to make an informed decision about their situation. So that's that. Do we have any questions before we take a break? Okay. Let's take a break then. We will come back at let's say 1:35, okay? 1:35. And then we will do more. See you guys in a bit. Right, so here are some concept check questions you can try on your own when you have time. All right, now on to the second half of the lesson. We're going to be looking at some genetics problem, and talk about how genetic information are actually passed on to the next generation. So much of what we will be talking about is based off of the work of this person. His name was Gregor Mendel. He's often called the fathers of genetics because of his work on um, finding out how genes are being passed on to the next generation. So some terms, some old ones and then some new ones for you. First of all, a gene, I just want to remind you, is a segment of DNA on the chromosome that encodes for a protein. In fact, you know how to decode the DNA now. You can take the ACTGs and then you can convert it into amino acids, right? Using the codon chart. Uh, but genes have different versions, right? Some genes have different alleles, different versions of it. For example, the gene for eye color could be brown eyes or blue eyes. That's the example I keep on using, right? And some genes have multiple alleles, more than two. Right? For blood type, for example, the ABO blood type has three different alleles. But even though there are multiple alleles that exist in the population, at most you can have up to two different ones, okay? Because we are diploid, we only have two sets of chromosomes, we only have two slots for them. So homologous, sorry, the there are two new terms here, not homologous. This is homozygous. Okay, homozygous or pure refers to individuals who have the same allele for a particular gene on the chromosome, whereas heterozygous or hybrid means the gene version on the homologous chromosomes are different. Let me show you a picture. It will make much more sense. Here we have two homologous chromosomes. And homologous chromosomes are same length, same size, and they have genes that are in the same order. Uh, but the gene version, the allele, might or might not be the same depending on who your parents are. So for gene A, the person inherit capital A from the mother and small a from the father. So for this particular gene, we say the person is heterozygous. Okay, or we can say they're hybrid. And over here, for gene M, they are both lowercase m, and so the persons for gene M, we say they are homozygous, okay, or pure. Can you find me another homozygous gene in this picture? Another homozygous gene. Yeah, H is homozygous, right? It doesn't even have to be lowercase, right? It could be capital case, capital case. These would be homozygous or pure, pure. Okay? So basically, homozygous means the same allele. Heterozygous means different allele. Right? And whether you are homozygous for one gene has nothing to do with whether you are homozygous for another gene. Okay, they are all independent of each other. All right, some more terminologies. A character is a heritable feature, something that can be passed on to the next generation. Okay, like the color of flower. Right? Heredity is not something that only applies to animals, right? Like plants, they can pass on genetic information as well. Okay, so a heritable feature is something that can be passed on. Not everything about you can be passed on to the next generation. So if you dye your hair green, your baby's not gonna necessarily, well, they will not. Okay, they will definitely not be born with green hair. Okay, if you work out a lot and you have very strong muscular body, your child is not necessarily going to grow up and have the same, right? So features that you've acquired throughout your life, those are not called character. They're not heritable because they don't have a genetic basis to it. Characters can have different varieties. Okay, if we're talking about flower color, then there are different colors that exist, and those would be called the trait. Okay, so a given character can have multiple traits, and these traits are actually caused by different alleles for that particular character. So let's say big B is the allele for brown eyes. Okay, and small b is the allele for blue eyes. So if someone is homozygous, big B, they would have brown eyes. That's not difficult to understand. If someone is homozygous, small b, then they would have blue eyes. The question is, what if someone is big B, small b? What if they inherit the brown eye allele from one parent and then the blue eye allele from another parent? Now they are hybrid big B, small b. Then what kind of eye color are they going to have? Well, it turns out to answer that question, we have to understand dominant versus recessive. A trait is dominant if it always cover up another trait. Okay? So in this case, brown eye is dominant, which we use capital letter to represent. it will cover up the blue eye allele. So even though you have a blue eye allele, it will not show because the brown one is dominant. So if someone is big B, small b, they will have brown eyes. Is that okay? So in this situation, two people can have brown eyes, but their genetic makeup are going to be different. However, if you have blue eyes, then for certain, your genetic makeup is small b, small b. There are no other choices, right? So this picture shows you a bunch of traits are dominant recessive in humans. If you are able to roll your tongue, then it's a dominant trait. If you cannot roll your tongue, then it's a recessive trait. So people who can roll their tongue, they are either dominant, pure homozygous, big R, or they might be big R, small r. There's no way to tell unless we, investigate more about their family history. But if you cannot roll the tongue, like I cannot roll my tongue, then for sure I am small r, small r. Okay? So two people who cannot roll their tongue, small r, cross means to have babies with someone else who cannot roll their tongue, then the whole family will not be able to roll the tongue, right? Because all they could pass on is the non-rolling gene. Okay, there are no big R in the family, therefore it cannot be passed on. Whole family is going to be like that. So following that logic, if you have one of these recessive traits and your partner also has a recessive trait of the same kind, then all your kids are gonna have that recessive trait, right? So if you like straight hair, hairline rather, straight hairline as opposed to Wiggle peak, and you want all your kids to have straight hairline, then all you have to do is find someone else who also share that trait, right? And it's guaranteed your child is going to have that trait, right? That essentially is what pure breed pets are like. Okay? Pure breeds means they will always preserve the trait if you breed it with another pure breed, right? That means they're all homozygous at all the locations, and therefore you are able to preserve the desirable trait. Okay. Um, so yeah, this is just an interesting thing for you to look at yourself, right? One other interesting one that is not here is did you know like five fingers are actually recessive, but six fingers are dominant. Okay. Some people have a genetic mutation in a gene called the Sonic Hedgehog gene, like the cartoon character, and that sometimes causes them to have an additional finger. Right? It's usually not like a full finger; it's more like a little appendage. But people usually get them removed. So if you have five fingers and you like all your kids to have five fingers, then all you have to do is find someone else who also have five fingers because that's a recessive trait, and recessive traits in both parents are going to be preserved in the family. That's the key takeaway. All right, so more terms. Here we have genotypes. Genotype is the genetic makeup of the organism. So like are you big B big B? Are you big B small b? Or are you small b small b? That's the genotype. You cannot always tell the genotype of a person by looking at them, okay? Unless they have a recessive trait, then you know they are going to be homozygous for that trait, okay? If they don't have freckles, then they are little f, okay? But if they have freckles, you actually don't know if they're big F, homozygous, or big F, small f, right? You know they have a big F, but you don't know what the other half of the genetic makeup is. So the manifestation of the genotype, the actual appearance, is called the phenotype. Two people with the same phenotype can have different genotype. Okay. Both of them have brown eyes, but one is big B, the other is big B, small b. Again, you wouldn't know unless you do a little bit of investigation, right? You study their family tree a little bit, then you might know afterwards. Okay. Do we have any questions before we move on? All right. So all these things are based on Mendel's experiment on peat plants. Peat plants. They are cheap, they are fast growing, and you can cross them easily. You can breed them easily. Okay, you can self breed them. You can breed it with other plants. You just take a brush, pick up the pollen, and then touch the other flower with it, right? And you can basically breed them quickly and easily. And he basically did a bunch of experiments with the peat plants and found out there are a set of traits that are dominant, and then there are a set of traits that are recessive. So how does he figure out which one is which? Well, I'm gonna go through one of such experiment with you. But first, he has a law that's called the law of segregation. I'll read it to you first, and then I'll explain to you. During the formation of gamete, each gene separates from each other so that each gamete carries only one allele for each gene. Basically, it means if there's the chromosome, and if one chromosome has a big Y for yellow and then small y for green, even though there are two alleles for the color of the seed, when you produce the gamete, these will separate, and one gamete will carry the big Y, and then the other gamete will carry the small y. Okay, that's what the law of segregation means. Okay, which is not surprising because meiosis causes your genetic material to be reduced by half, so you cannot have both. You can either have the big Y or the small y, and that's going to be passed on to the next generation. All right, let's take a look at this thing here. We will do it in the class notes so I can post it for you afterwards. Okay. So the original generation is called the P generation, the parent generation, basically. And Mendel took two plants. Okay? One plant has round seed, and then the other has wrinkle seed. And he knew, or he assumed they are pure in that he observed the same plant for many generations, and he noticed these plants always produce round seed. And also, he observed the wrinkle seed plant for many generations and noticed they always produce wrinkle seed. So, based on that, he can reasonably conclude that the plant is pure. Okay, it's big R for the round seed, and then it's small r for the wrinkle seed. Okay, now this is confusing because like how does he know the round seed is dominant, right? He didn't, okay? He didn't know whether round is dominant or wrinkle is dominant. But then, like I know because you know we learn about it, right? Okay, so I'm gonna put big R for the round seed and small r for the wrinkle. But just hold on, okay? Let's take a look at the results, and then hopefully you will understand how he found out. All right. So look, the F1 generation is the first generation that you get from the parent generation. So if you take a look at this round seed here, it's big R on both chromosomes. So half the gametes will have the big R, the other half will also have the big R, right? All the gametes are going to be big R. For the wrinkle one, all the gametes are going to be small r. So this thing here is called a Punnett square. Punnett square. Punnet. Punnett square. Okay. And we use a Punnett square to try to figure out the probabilities and percentage of what kind of offsprings you're going to produce. So here, if the big R combines with the small r, then the offspring will be big R small r. Similarly, this and this combined will get big R and small r, and so on and so forth. So in the end, you can see 100% will be big R small r, and 100% will be round seed. Okay. So he didn't know the round is dominant when he did the experiment, but then when he looked at the result, he can conclude the round is the dominant because the wrinkle completely disappeared. Okay? If you take a round parent and a wrinkle parent and you cross them and all the offsprings are round, then you can conclude the round is dominant trait because it covered up the wrinkle trait. Okay. So similarly, if he takes like a yellow seed, yellow plant, yellow seed plant, and then cross it with a green seed plant, the green ones are the ones we are familiar with, right? Like when you eat your frozen peat or something, they are all green, right? And so he crossed them, and then 100% F1 are yellow. Then which one is dominant? Is it yellow or is it green? Well, the green disappeared, right? The yellow covered it up. We can conclude yellow is dominant. Do you understand? Okay. This only works if both of the parents are pure, though. Right? And how did he know the plants were pure to begin with? He observed them for many generations. Right? If the plant always produced yellow seeds, then we can assume it is a pure yellow seed plant. If the plant always produce green seeds, we can assume it is green plant seed. Okay, is it always accurate? Not always, but reasonably accurate. Okay, so that's the F1 generation. Now we're going to take a look at the F2 generation. The F2 generation is what you will get when you take individuals from F1 and then self-pollinate. Okay, plants are also useful in that regard that you can do asexual reproduction, like just cross their own seed with their own eggs. And so from F1, they are big R and small r. So over here, it doesn't matter which seed you put where. In meiosis, the alleles will separate. One gamete will have the big R, the other will have the small r. Similarly, big R and small r. So if we combine these gametes together, big R and big R, you get that. This and this will give you big R, small r. This and this will also give you big R, small r. We typically write the big one before the small one, okay? And then small r, small r. Let's take a look at the genotype. These are the offsprings, right? There will be 25% chance of big R, because it's 1 out of 4. What about big R, small r? What's the probability? What's the percentage chance for big R, small r? Anybody? 50%. That's right. 2 out of 4. And then another 25% small r, small r. So these are the expected genotypes, the genetic makeup. For the expected phenotype, okay, what are these ones? Are they round or wrinkle? Okay, because it has the capital R, it is going to be round, right? Yeah, and these are also round. So 25 plus 50 is 75% round. And then 25% wrinkle. Okay, so that's the expected outcome. If you produce 100 offsprings in F2, you would expect roughly 75 of them to have round seed and roughly 25 of them have wrinkle seed. It's not going to be exactly 75 and 25, right? Because these are probabilities. It's like if you flip a coin 10 times, you are not necessarily going to get five heads and five tails, right? But it will be close. So that's all for today. Does anyone have any questions about anything we talk about today before we go? All right. We'll do more on Thursday. Please print the study guide if you haven't already. Enjoy the rest of your day. See you later.